Hi. Thank you, first of all, to OHBM organisers. This is always my favourite conference of the year, and so thank you for giving me this opportunity to speak today. I'd like to talk about how shifts in myeloarchitecture profiles characterise adolescent development. Myeloarchitectonics is the pattern of intracortical myelin that you see. So here on the left, you can see myelin sheathing this horizontal axon collaterals. We often think about myelin as being very important for conduction speed, but when we look at the intracortical area, these fibers are actually so short that myelin has a different role to play. It actually imposes a spatial structure upon these axons, so it more governs information flow in the cortex. And as early as 1894, I've seen that Flechsig documented that there was differences in the development of intracortical myelin. So you see in that densely spotted green areas, these primordial sensory areas that develop very early. Then there's the next surrounding regions, those intermediate areas. But there's large swaths of the association cortex that continue to develop well into adolescence. And histological studies have continued to suggest this, and specifically have shown that this is a depth-dependent process that occurs. So we wanted to probe this adolescent maturation of myeloarchitecture using this really wonderful data set out of Cambridge, the Neuroscience of Psychiatry Network. And specifically, we've decided to look at the magnetization transfer data, which it's possibly one of our best markers of myelin for in vivo imaging. And the data set is an accelerated longitudinal design. So for each subject, we use 2 time points, and by nesting these individual trajectories into a larger linear mixed effect model, we can better build a model for how the changes occur across the entire age range from 14 to 27. So we're very interested in these depth-dependent changes. So we operationalize this by building 12 equivolumetric surfaces between the peel and white matter surface. And then we sampled the MT intensities at each of those surfaces at every vertex. And then we parcellated that into 1,000 nodes. And here you see some group representative profiles. So I'll always present these with the y-axis as cortical depth. So peel is at the top, and the x-axis there will be the MT intensity. We first wanted to work out whether what we get from in vivo imaging was similar to or recapitulates what we see from the histological data as well. So we looked back to these really beautiful images that I liked from the FOTS, and we translated these hyperstained myelin sections into these histological profiles you see. And then we looked at the comparison between these post-mortem profiles with the in vivo data we had. We were really inspired by Axel Schleicher's work and Carl Zillis on this quantitative profile analysis. So they highlighted that you could use the central moments of a profile to characterize it in a more simple way. And specifically, we looked at the mean and the skewness here, so the first and the third moments, for just interpretation this was easier. And we see that in both the postmortem and the in vivo data, that the mean of these profiles is highest in sensory areas and drops off as you move to paralimbic. So here you see in the blue are these sensory areas, yellow is unimodal, pink is heteromodal, and the green is this more paralimbic regions. The skewness actually moved in the opposite direction. It increases from sensory to more limbic. And the skewness refers to this non-linearity of the profile. You see a very positive skewness when there's a preferential accumulation of myelin towards the white matter boundary. So you can see that skewness quite nicely in those green profiles there. So we move this on to look at the whole brain as well, and we see that the mean, once again, it decreases from sensory to the more polar regions, but skewness has a different pattern where it is highest in the limbic ring you see there, and it drops off as it moves to sensory. And you see that by the distribution of values across these levels of laminar differentiation at the bottom. And really crucially, what we found was by using these central moments, we could capture depth-dependent changes in a way that we don't have to test each surface, because if we were to do that, you'd have a non-independence of the surfaces that's very difficult to deal with in statistics. So the central moments give us a very simple way to deconstruct these profiles. We found using linear mixed effect models that the mean increases in these more lateral association areas across the age range, and you see that with a rightward shift of the profile. And there's a different pattern that emerges with the changes in skewness. You see significant decreases in skewness across the age range, across a broad range of cortex as well. And you see there's a preferential accumulation of MT intensities, or most likely myelin, in those mid to deeper layers of the cortex during this age range. Because these patterns seem to be very unique, we hypothesized that they were reflecting different biological processes. We tried to tap into that by using this wonderful Allen Brain Atlas dataset to correlate the spatial maps of age-related change with the gene expression maps. Specifically, we found for the age-related change in skewness, it was significantly associated with 86 genes, and these genes were enriched for oligodendrocytes as well as adolescence and young adulthood. So this seems to be gauging that what we see, these age-related changes in skewness, are specific to the changes in oligodendrocytes. So it's a process of myelination. And this was robust when we also controlled for changes in cortical thickness, because we know a lot of changes in morphology is happening at the same time. But this seems to be distinct or unique from those morphological changes. And really for us, this drove home that because the only significant gene expression relationship we found was with this age-related change in skewness and not the baseline maps or age-related change in mean, it's very important to capture these depth-dependent changes when we're thinking about dynamic biological processes. We also decided to look at a more large-scale reorganization of microstructural similarity during this age range. And we turned to using the microstructure profile covariance, which is a technique we recently validated in histological and in vivo work. And this simply takes those MT profiles I showed before, and we perform a partial correlation between these to capture the microstructural similarity between two areas. And then at an edgewise level, we measured age-related changes in microstructural similarity. At first, the pattern we were seeing was very maybe obtuse. We didn't really know what this distinct pattern was. So we decided to use a nonlinear dimensionality reduction technique called diffusion map embedding to see what axis of changes is occurring here. So we found that the principal component explained 36% of variance in this T statistic matrix. When we plot that back onto the cortical surface, you see areas that are very similar color on that brain map are changing to become more similar and then becoming more dissimilar from the areas that are different color. So you see sensory areas, those purple areas are becoming more similar to each other and then becoming more distinct from those more limbic areas in yellow, which are becoming more similar to each other. We can see this in another way if we break that gradient down into 10 discrete bins. We take that colorful matrix up the top right there, and we break that down into 10 groups, and we look at the average profile within each one and perform a correlation. You see that there's overall a very similar pattern in the youngest age strata and the oldest, but there's one very distinct change you can see there in those correlation matrices there. Early on in adolescence, it seems there's some profiles in the middle that seem to bridge between these two modules, but there's a really clear segregation as you move into later adolescence and young adulthood. We decided to break this down more by building these microstructural profile covariance matrices within the youngest and oldest age strata. And once again, we use diffusion map embedding to see the axes of differentiation this occurs on. As you'll see, these axes are very similar to what we see of age-related change. So this led us to the hypothesis that if there already exists this axis of microstructural differentiation from sensory to fugal, and then the age-related changes occur on the same axis, that would lead to an expansion of that sensory-fugal axis. And we tapped into this more so by looking at in what regions this occurs. So as you see, this age group difference map shows that the prefrontal areas are becoming more similar to that more sensory anchor, whereas these blue areas around the Sylvane fissure are becoming more similar to the limbic regions. You can see this once again if we were looking at the distribution of gradient values as well. So in the top area versus the bottom area, I'll highlight those yellow and pink association distributions. You see it becomes much more bimodal as you move into young adulthood. And we see that specifically this is because the central areas of that gradient, those association areas, are being drawn outwards. They're moving towards one of those anchors. So the biggest effect sizes in terms of changing gradient values is happening at the center of that gradient. And of course, this all moves back to those changes we're seeing in the myeloarchitecture. So myelination during adolescence, which is driven by all this learning and experience that's happening, it's reshaping the myeloarchitecture, and it's also restructuring that local connectivity that we see. But this also impacts a more grander cortical landscape of the microstructural patterning. So specifically, our findings show that this depth-dependent myelination is driving this segregation of these lower order areas from more higher order areas. And we're really excited about delving into this deeper with this multi-scale model to understand the functional and cognitive consequences. So I'd just like to thank my wonderful team at the M&I, and especially my mentor, Boris Bernhardt, as well as our collaborators at Cambridge, especially Richard and Ed. And I'm doing a poster on this on Thursday, and I'm also having another poster on a cortical wiring scheme called deep embedding. So please come by and have a chat. So thank you.